Hey everyone, we're going to continue chapter 6 looking at enzymes. Now, in our past chapter, we talked about what enzymes are, kind of classifying the way in which they act. We talked about their catalytic mechanisms. But today we're going to talk about the field of biochemistry called enzyme kinetics, which is the study of the rate at which enzymes specifically react. So this is a huge area. And again, this area of study is very important because it allows us to understand what can affect enzyme reaction rates. It lets us look at how drugs and other substrates and ligands affect enzyme rates and their reactions and products. So again, huge area. Generally, the enzyme kinetics are affected by things like the enzyme itself. So its rate is going to be determined by the enzyme. The substrate that an enzyme is acting with will also, of course, affect the rate of the reaction. Any other effectors, so these can be other substrates, other inhibitors, and we'll talk about those in this chapter, will of course potentially affect the rate of an enzyme. And a big one that we're not going to focus on as much, but is still very important, especially when you're thinking about planning experiments, is the temperature. Temperature will potentially speed up the rate or slow down the rate depending on what your temperature of a reaction. And so they can affect enzymes. Since enzymes are catalyzing a reaction, temperature is very important. So before we head into kind of graphically looking at enzyme kinetics, I first want to go over the general idea of the formula we'll be discussing for enzymes, which is, in a very simple case of enzymatic catalyzation of a reaction, you have an enzyme and you have a substrate. So again, some protein that catalyzes a reaction, and whatever the molecule, protein, peptide, nucleic acid, whatever it is that actually is going to bind the enzyme for the reaction. These form, remember, our temporary transition state. And that's because the enzyme helps to stabilize the substrate in this transition state before it forms the product. So we have our starting enzyme and substrate. They go on to form a temporary point in which the enzyme is bound to the substrate. The enzyme does its work. It helps the chemical reaction proceed, helps catalyze the reaction, and then you have release of the product and you have the enzyme back to normal. So these are the states that we're going to be talking about. Just keep these in mind when you see these letters E and S. And if you see ES together, that means enzyme when it's bound to substrate. These are common formulaic ways of expressing enzymes and substrate reactions. So most of this work in enzyme kinetics was done by two scientists, Maud Menton and Leonor Michaelis. So you're going to be seeing, and we're going to be talking a lot about Michaelis-Menten reactions, Michaelis-Menten enzyme kinetics, and Michaelis-Menten plots. I just wanted to highlight these two, especially to give a shout out to Maud Menton, who was a prominent female scientist. I think female scientists need to get all the recognition that they deserve, especially in situations where we have females underrepresented in the sciences. So Maud Menton is where the Menton comes in Michaelis-Menten. So again, why do we study enzyme kinetics as a whole? Why is this such a big field? Again, it gives you quantitative descriptions of the biocatalyst. So you're going to get an actual number that is related to how well it does its job at catalyzing a reaction. We can also help determine the order in which things bind, which might not seem all that important when you just say it in this way, but if you're trying to figure out the mechanism of something happening in the body, you want to know how things are reacting, the order in which they're reacting. Which complex do we need to target at what time? So this order is actually a very, 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 very important thing when it's, again, related to human medicine and drug development. Binding order and the mechanism of action, which we say the way that things are happening biologically, can be determined from enzyme kinetics. We can also find very effective inhibitors. Sometimes we want to inhibit things. Sometimes we want a substrate that is going to mimic a normal substrate of an enzyme. So being able to understand how the rate of an enzyme changes if you have an inhibitor, if it is an effective inhibitor or not. These are important. So again, I gave an example in our live session about looking at an inhibitor for the COVID, the binding of the COVID-19 spike protein. And I, based on data, said, do you think that this would be a good inhibitor for that spike protein? So this is the kind of data that you can get from enzyme kinetics.